I'm sure many of you have heard the word CRISPR before, but what you may not be aware of is that hidden behind this catchy acronym are an endless array of CRISPR subtypes and modified variants. Some of this variation exists naturally, and some has arisen out of the clever modifications made by scientists. In this video, I'll do my best to give you an introduction into just some of this tremendous diversity, as we discover how just a small tweak in the design of a CRISPR system can repurpose its function entirely. Before we jump into these more abstract creations, let's make sure we actually understand the original. The acronym CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. Let's just leave it at CRISPR. As the name suggests, these are genetic structures that consist of a series of repeated DNA sequences separated by variable parts known as spaces. A seminal breakthrough made by Mojica and colleagues in 2005 demonstrated that these spaces derive from and confer resistance to other genetic elements such as bacteriophages and plasmids. This whole section is preceded by a leader sequence rich in DNA bases adenine and thymine. Adjacent to this are genes that encode CRISPR-associated proteins, and in some systems a molecule known as transactivating RNA. Following transcription and translation of the Cas proteins, these components assemble into a complex capable of cutting at a particular sequence of DNA. The transcribed spaces, once processed from their pre-CRISPR RNA stage, act as the guide for an endonuclease enzyme, such as Cas9. This guide RNA, along with the protospacer adjacent motif just downstream of the target DNA sequence, are what give CRISPR-Cas systems their specificity. In their 2012 publication that culminated in a Nobel Prize, Doudna, Charpentier, and colleagues showed that the sequence of the guide RNA can be programmed to cut at a desired location within a genome, thereby unveiling its potential use as a gene editing tool. Now that we have a good understanding of what a natural CRISPR system looks like, let's take a look at some cleverly engineered versions that have modified activity. A somewhat drawback of regular CRISPR-Cas9 systems is their inability to induce fine-scale mutations, such as a single base point mutation. To circumvent this issue, scientists have created so-called base editors, such as ABE, the adenine base editor. This system is capable of converting a single AT pair into a GC. Let's take a closer look and see how this works. The deamination of adenine yields inosine, which is read as guanine by DNA polymerase due to their structural similarity. However, prior to this study, no enzymes were known to deaminate adenine within DNA. The researchers took a tRNA adenine deaminase and mutated it to be capable of operating on DNA. They then fused this enzyme to an altered CRISPR complex known as dCas9, which carries a defective Cas9 protein unable to induce double-stranded breaks. It is, however, still capable of locating and binding to the target sequence. dCas9 can therefore bring its fused adenine deaminase to the target site, where the change from an A to a G can be catalyzed in a specific region of DNA. Now you might be sitting there wondering why anyone would even want to do that. Well, it turns out that there are a significant number of pathogenic mutations that comprise the modification of only a single base. Base editors, such as ABE, could allow us to rectify these disease-causing variants in a safe and reliable manner. So, what other purposes can CRISPR systems be adapted toward? Let's take a look at another scenario, this time involving regulating transcription. The dCas9 variant mentioned earlier has been extensively used as a repressor, where it can bind to genes or their promoters to inhibit transcription. Amazingly, scientists have also figured out a way to turn this CRISPR complex into an activator, where it increases target gene expression. To do this, they fused the omega subunit of RNA polymerase to a defective Cas9 complex. The introduced guide RNA molecule is designed such that it escorts the polymerase right to the perfect place on the promoter to commence transcription. Synthetic biologists have also shown this to work in regulating the expression of gene circuits in yeast for metabolic engineering purposes. Typically, expression in these settings is modulated through the use of various elements like the TET and LAC repressor proteins, which assist in switching on or off different genetic pathways as desired. Assembling these circuits, ensuring that they are orthogonal and tunable, can be a complicated and laborious process. In this study, the researchers expressed the dCas9 fused with the RNA polymerase omega subunit inside of yeast. They then sequentially induced expression of a range of guide RNA molecules that targeted the regions upstream of different promoters. They were able to show corresponding spikes in expression of the desired genes, proving these CRISPR transcription factors to be effective modulators of synthetic gene circuits. It seems there really is not much these malleable tools can't do. Given just how many different functions have been realized through repurposed CRISPR systems, it is truly astounding that it has been little over a decade since CRISPR gene editing was first discovered. Although we only got to see a couple today, I hope this video has given you an appreciation for the enormously widespread potential this technology holds.